He's a psychologist originally in Wisconsin and then trained in neurophysiology, and his career has been focused on understanding the mammalian brain, and particularly the sensory motor aspects of the mammalian brain and their evolution, using techniques in anatomy, electrophysiology and brain imaging, I think. Jon is very well known neuroscientist. He's published a great number of papers and has been very influential. An interesting program on the internet is NeuroTree, and it's interesting to look there and see the links between different neuroscientists. It shows you who was the graduate student with whom and who were the people that they trained, and then who were their scientific children. And if you look at Jon on NeuroTree, you see that his background goes back to a very important group of comparative neurobiologists in Wisconsin that included people like the Woolseys and Wally Welker, and his own academic children include people like Leah Krubitzer, who's been here, many other people that I think are keeping alive this comparative approach in mammalian neurobiology. And it's really important because there's so much focus now on just one or two model animals like the mouse. But there are so much variety in the nervous system of animals, and particularly in mammals, that if we just focus on these one or two focus strains, then we're in danger of really missing out on much of the richness of animal nervous systems. And so I'm really delighted that Jon is here. We had Leah talk last year, and this year Jon is going to give us his perspective on the evolution of the human brain. Jon.
Thank you. I really feel fortunate to be able to come here. It's been a while since I've been in Barcelona, and it's a great time, and it's very nice to be able to talk to you. The question is that why are we, or why am I especially interested in the evolution of the human brain, how that came about? And it's partly because if you reflect on what the human brain is capable of, it's so amazing and so impressive. And this is, you can think of all kinds of examples of scientists, but you can think of writers in literature, of artists of various sorts, of athletes at the Olympics, your own children growing up and acquiring abilities and so on. And I'm continually amazed at the range of possibilities of human behavior and human skills that is all made possible by a somewhat highly similar brain that we all possess. And yet, in that framework, our Nobel Prize winner Francis Crick and Ed Jones, who just recently died, one of the great anatomists of our time. Yes. Only a few. Yeah. Well, he died at a meeting while he was talking. So he died doing what he liked to do best. They said we have to develop new techniques to know more about brains. We have to know more in a comparative sense about brains. What we know is strikingly little. And that's going to be a little bit of what hampers me in the talk today. But as you know, imaging studies of human brains and so on have led to a lot of new knowledge in a very short period of time. Now, we have a past, and we have potential ancestors from the past, and they existed at various times in the past. Here's some sort of a scale and some examples up to modern Homo sapiens, and something about their brain weight, and I think fairly reasonable estimates, and I can get to you how we can do these estimates, of the numbers of neurons. And one of the features of a human brain that is so impressive is that we have more neurons in our brain than any other life form on this planet, including whales with brains 6 times larger, elephants with brains 4, 5, 6 times larger, and so on. We still overmatch them in numbers of neurons by a good margin. So neurons being the basic computational unit of the brain, that's maybe why we have such an advantage in propagating ourselves, changing the environment, and in a sense, ruling the earth. Now, people have always been curious about their origins. And so every culture has at least one story about where people came from and how they emerged. And being from Tennessee, where it was once illegal to teach evolution, I'm sometimes required, no, I'm not really, to teach the Genesis story of Adam and Eve as our beginning about 6,000 years ago. But it's more intriguing for me to think of other cultures and what they might say about our origins. And here's from the Maya Indians of Central America. And their story was more intriguing to me because the earth was inhabited by gods, but not by people. And the gods were dangerous and they kept people from inhabiting the earth. They always existed as spirits in the Milky Way, but they couldn't come to earth until the gods had put the heads of other gods they killed up into this tree, and this other goddess came along, and one of the heads spit in her hand, and she became, this is why everyone should be careful, spit in her hand and she became pregnant and had twins. And the two twin brothers then killed all the bad gods through a wonderful trick. I don't know how they did it, but one brother cut the head off the other brother and then put the head back on, and he came back to life. And the other gods, the bad gods, were so impressed with that they all wanted their head cut off. And he cut off all their heads, and that was the end of them. And people could come from the Milky Way to Earth and start to populate the Earth. But we have another story, and the story from science is that we came from a series of ancestors that in the past, greater times in the past, were progressively less like ourselves and more like something else. This would be what our ancestor, this is a famous skeleton based on that Lucy, 3.2 million years ago, and roughly what our ancestors would have looked at that time. And if we go back in time and just look at skulls, it's pretty obvious something is changing the shape of the skull, and the brain case is getting smaller. So our large brains came from ancestors with much smaller brains, and if we go back to the breakpoint between ape lines and human lines, the chimp-human break, about 6 million years ago, our brains were about 1/3 or less our present size, and roughly equivalent to a chimpanzee's brain in size. But this is a human brain, happens to be a former graduate student of mine. And you can see his brain, like most of the brains in this room, fill the skull rather tightly. There are no missing parts. He's an assistant professor now. But this makes the point that you can get brain size estimates fairly accurately for many mammalian brains just because the brain fits the skull cavity very tightly, or the skull cavity, if you take it adjusted to the size of the brain. And here's a comparison between a human brain, marvelous thing, and a chimpanzee brain of roughly 1/3 the volume. And that's what a brain of our ancestors would have been like 6 million years ago. Now, we don't know, although we're rapidly learning, that much about the organization of the human brain, and we don't know that much about any brain, but this is a model of visual processing in a flattened macaque monkey brain that was done by a former graduate student of mine when he was doing his postdoc with David Van Essen. And this is the one that appears in all the textbooks when they're talking about the visual system. And the real point here is that I'm going to emphasize talking about cortex or neocortex, as we would say, in talking about human brains and other animal brains, because it makes up about 80% of our brain. And so it's a very important part of the brain. And this macaque-monkey brain, with the cortical areas all laid out here, is an approximation of the organization of a macaque-monkey brain. So there are a lot of debates about the accuracy of this depiction. And I could go into a lot of the arguments. But that's not the point. The point is that these colored areas all have an important role in processing visual information. And it roughly estimates the number of visual processing areas that a macaque monkey would have. And it's roughly 35 areas. So if you wanted to say 40, I'd say fine. If you want to say 25, I'd still say fine, because there's that much uncertainty. And in fact, in this paper, they rated these areas on the basis of 1 to 3. 1, we're really sure about this. 2, not so much. 3, pretty much of a guess so far. And they only gave 5 areas a 1, and 2 of them I disagreed with. But what they were able to do on the basis of looking through the literature is come up with a wiring scheme for this kind of system. And again, there are so many uncertainties, and there have been arguments and revisions and so on, because this is 20 years old. But it gives an idea of the magnitude of the area by area wiring diagram, the complexity of it for processing visual information. And there's more complexity than you would even see here, because we're not talking about subcortical interactions with the cortex. This is just a cortical scheme, and it's not talking about modular subdivisions within the cortex, so it's more than that. And it's emphasizing a subdivision of the brain. You would say a nucleus or a cortical area. And these have a special feature of representing, say, all of the visual field, or the hemifield for one hemisphere. So each one of those areas. So they're doing something for all of visual space. And this concept of a cortical area goes way back in time, primarily to Brodmann, who called them, these are the organs of the brain, like a kidney, like a liver, and so on. Each one is doing something a little different, and a lot of it, what it's doing, depends on the inputs, and its functions depend on its outputs, but each has its unique sort of internal organization to do those tasks. And here's one that we published a few years ago with a graduate student of mine, Troy Hackett, who's now a faculty member at Vanderbilt. And this is just early processing stages in the auditory system. So we could do this for the auditory system, we can do this for the visual system, we can do it for somatosensory system. And in primates, the visual system is clearly the most complicated. But these other systems have quite a bit of complexity as well. So we can say that if this is a macaque monkey, and roughly the cortex as a single sheet spread out in a human is about 8 to 10 times larger, you can imagine that we could scale the complexity of a macaque monkey brain up maybe by 8 to 10 times. And so the complexity is sort of mind-boggling.
Who generated this diagram, John?
So my graduate student and I did this for a symposium some years ago on the auditory system. Now, early views of how human brains evolved, and this is from the Thompson textbook, which is the first really good textbook in physiological psychology, as we called it. Now we would say it's neuroscience. But it shows that basically, it reminds me of the maps that you saw of the Earth, where you had unknown areas, whole areas of the Earth, monsters be here, things like that, but nobody knew what was there. And so basically their conclusion was if you did comparison studies, we had some knowledge of what most of the cortex in a rat did, and practically no knowledge of what a human brain, most of the cortex in a human brain did. And you just said, well, they got more association cortex. Association cortex was, another word for it was cortex under anesthetic that's non-responsive to stimuli, so we don't know what it does. Must be associating things. And here's another view, and this is a view of Paul MacLean from NIH, the triune brain, and I don't know if anybody ever read that book, but basically he says we have embedded in us, this is really just straight out of Freud, a reptilian brain, a sort of early mammal brain, and then the new mammal, current mammal brain, all imposed at 3 different levels doing different things like the id, the ego, and the superego. And the trouble with this is that when you change one part of the brain, you have to plug it into other parts of the brain, and everything is sort of changed. So you don't really have these separate domains. My former collaborator, John Allman, wrote a book on brain evolution, and his analogy was that he went to the power station in Los Angeles, and in the power station, they had some of the latest equipment for running things and some of the oldest equipment for running things, and they were all integrated together. And the reason that they had this old equipment is that you couldn't really shut off the power and say we're going to take a few months to redesign everything. It has to run all the time. So you tinker with it and change and add and whatever. And that's how brains evolve, because you can't shut them down and say, this is really a bad start. We're going to go reboot here and redo this. And so I want to talk about cortical organization in early mammals, because that'd be a starting point. We could push back as far as you can imagine to single cell life, but it wouldn't be very practical to do so. And we do so, we're sort of wandering around looking at relics of the past and imagining what they might be. This happens to be in the Natural History Museum in Paris, a wonderful place. And I'm going to emphasize the neocortex again because it's become such an important part of our brain. And it looks sort of, if you cut through it, it looks sort of homogeneous. This is in a primate, prosimian primate, but we've settled on dividing it into 6 layers. Different people have made different numbers of layers because they become sublayers in other schemes. But 6 layers because the 6 layers have different inputs and outputs and types of neurons. And so they have different functions. So one of the great things about neocortex is that it's made up of excitatory and inhibitory neurons that interact, neurons that project to distant targets, and neurons that project to other areas of cortex, and neurons that are intrinsic and don't project outside of the region. And this turns out to be a tremendously useful kind of organization because it can be modified and enhanced in so many different ways. You also have these sort of modulatory inputs that come in from dopamine systems and so on that change the circuitry and its functions over short periods of time. And then you have longer changes that can take place over greater periods of time that allow this to be a system that can learn in some sense, or be modified, or be plastic. Tremendous variability and potential for the cortex, and we were lucky to get it. And this just shows one of the features is if you make a little injection in this cortex, you can see everything that's being connected here. And the real point is that neurons in a column or a vertical array across the layers are highly interconnected. And then you have outputs, you have lateral connections to nearby groups of neurons and so on, and projections that go subcortically to other areas of cortex and so on. But the vertical interrelationship of neurons in a column is one of the essential features of processing in this structure in all areas of cortex, and it gives you some amazing things that you can do. And one is you can reiterate. You can do some simple calculation processing in one area of cortex, send it to another area, repeat the same kind of steps, and get something more sophisticated or more advanced every time you repeat the step. But you can also divide up into different lines of processing and come up with different outcomes, and then converge different outcomes to complete something quite new. Well, this is all things very similar, I mean, familiar to you probably, but it's, what would I say, cortex made us great. And it's because the potential of this cortex to do so many different things. Now, where did it come from? Best presumption is it came from something like the dorsal cortex of reptiles. We really don't know exactly what it was like, because reptiles, when they first emerged, now people in comparative say, well, let's not call them reptiles anymore. We'll say early amniotes, because they could lay eggs on dry land. So they have these membranes around their egg. Divided into two lines. So about 340 or so million years ago, these two lines separated, and one came about and led to present-day mammals, and everything in all the branches of that line are gone except mammals. The other line gave us all our present-day reptiles and birds. So we can go to the birds and we can go to the reptiles and get a guess about what the line that led to mammals was like, but there are uncertainties. But reptiles, this is a turtle cortex, have a great big olfactory structure up here. This is the forebrain. Then they have what I'll call hippocampal cortex, because it's the homologue, the same structure as our human hippocampus, and piriform cortex, which is olfactory cortex in humans and in all mammals. And so, and this is dorsal cortex, the homologue of neocortex, and it only has two inputs. Visual inputs dominate throughout here, and then there's a region of somatosensory inputs. Yes?
So I've heard people talk about the dorsal ventricular ridge.
Yeah.
The same thing?
No. Most of what would be incorporated in the dorsal ventricular ridge now would be called part of the basal ganglia, homologue of a basal ganglia. There are still a couple, and amygdala, so subcortical structures. And so you have to remember here that we have the dorsal cortex without any auditory input, for example. Yet these reptiles do quite well in the auditory realm. Their auditory inputs are going to the equivalent of the basal ganglia and the amygdala, and that's how they're running, using their auditory information.
So on what basis is the ventricular ridge now assigned to be basal ganglia as well?
So people have turned out that histochemistry turns out to be a great cue. Right. And so the people working on birds and reptiles met maybe 5-10 years ago, and this is amazing because it would never happen working with mammals. They agreed on a new terminology, and they said, we throw out our old terminology, we now know what the correct homologues are, and they came up with a new terminology. Anne Grable, who works on basal ganglia in mammals, I remember she said people working on mammals would rather exchange and use each other's toothbrush than their terminologies. You know? So people feel strongly, no, I have the right term for this.
Another question. If you now look at growth of olfactory bulb, the piriform cortex, how heterogeneous is this system that you move through it?
Is this piriform cortex homogeneous?
Compared to this olfactory bulb, if you go through this whole area you outlined there, because there's one line going around it, right?
Yeah.
Are these very heterogeneous subdivisions or not?
Here's a section through this kind of cortex from two different reptiles. And you can see that this medial cortex has the densest arrangement, and these are pyramidal neurons. This is fibers on each side. So in a row of pyramidal cells densely packed, it looks like a hippocampus in the sense that we see the same dense collection. This is the dorsal cortex up here, a scattering of pyramidal cells and other cells. Here's another one, the medial cortex, hippocampus, the scattering of cells over here, which is the dorsal cortex. And then this lateral cortex, which again is basically a single layer of compact cells, mostly pyramidal cells.
The cell types is fairly uniform, though.
Yeah, the cell types are,
The cell types vary.
all three of these areas, in a sense, are wired in a very similar manner. And the dorsal cortex here is wired in a different way than neocortex is. And basically, here's how it's wired. So here is a scheme, here's a section of the cortex. And basically, in the cortex, the input comes into a layer of intrinsic cells, layer 4. And it's processed very locally right there, and then sent to the other layers. In the dorsal cortex, or in the hippocampus, or in the piriform cortex, the inputs come across the dendrites of these neurons that are the pyramidal cells that are providing the output. So each pyramidal cell is affected by thousands of inputs and inhibited by local cells that also get connections and feedback connections from the pyramidal cells. And then the pyramidal cells send a subcortical output out, and in the dorsal cortex they also send to the medial cortex, which is the hippocampus equivalent, because they want to have some memory of this. So very simple, an input and output, and the purpose is simply to sum thousands of individual inputs and come to an output from that. So it's a good system for, like this cerebellum, for making adjustments, behavioral adjustments. This cortex is essential for the habituation behavior of a turtle to withdraw its head. When you push a balloon towards its head, it'll withdraw. But after a while, it'll habituate. This cortex takes that information and somehow changes the wiring so that the head response habituates out. So it's not good for making discriminations of fine details of the visual scene. It doesn't do that very well at all because it's got all kinds of inputs, each having very little effect. So the difference between neocortex and dorsal cortex and what it's doing and how it's organized is tremendous, and this is why I think we are so fortunate in getting this structure, because of its potential for doing so many different things.
Just a functional question. I was intrigued by using the word reflex. So the turtle withdraws its head reflexively.
I don't know if I said reflex, but it habituates to that response.
Right.
It habituates that withdrawal response and kind of learning. Simple. Okay. Yeah. And we know from the fossil record, so we can learn some things from the fossil record about any stage in mammalian history, and we know early mammals were small. They were mainly or almost exclusively nocturnal. They were running around being terrorized by dinosaurs. And, but they could hear at a higher range than any reptile. So they could communicate with one another in a range that the reptiles couldn't. Babies could call to their mother and so on. But they were small and they had little brains. And this is the mammalian radiation, and some starting about 250 million years ago. And the point is that you can sort of group them into 6 major branches: the monotremes, the marsupials, Afrotheria, this is the modern phylogenetic scheme here, Xenotheria, Eucontigleres, and Laurasiatheria, and so on. And we're over here in Eucontigleres. And so if we wanted to know what early mammals were like, we would say, what do members of all these branches have in common? This is sort of the standard cladistic approach to comparative deduction about the past, ancestry, bones, whatever, anything. you say things that are in common or shared widely are things that were likely retained from the past, and that rather than evolving independently. But you'll have little changes where things have changed and they're not recognized anymore, or they're considered new. And that's basically the method. From the fossil record, you don't know anything about the internal structure of brains. You only know about the size, the shape, fissure patterns, and so on. And you can make some reasonable inferences from those kind of features, but mainly you have to learn from comparative studies of existing mammals. And the wider you search, the more likely you are to reconstruct the probable ancestor accurately. Here's just an example of a skeleton. So you can see for size how small early mammals in general were. And here's, sometimes you could get a pretty good idea from this endocast what the brain was like. And this was one that's only 85 million years ago, but things really didn't start to change until about 65 million years ago when the dinosaurs became extinct. Big olfactory bulb, a lot of olfactory cortex, piriform cortex, a small cap of neocortex, the midbrain exposed and not covered by the neocortex. So those way early mammals had that kind of shape. Now, it seemed reasonable if we wanted to know what the brains of early mammals were like, instead of looking at an elephant or a whale or a human, where we know the brain has changed enormously, we get lost in trying to find common features of these animals, even if you could study them in great detail. So it seemed reasonable that we would go and look for present-day mammals that had small brains and little neocortex and say, well, maybe these animals have changed the least. But you have to go one step further and say, but the way they changed, even if they changed the least, might have been different in different lines. So we should compare across all the major divisions of the mammalian radiation, small-brained animals, and see what they have in common. And this is one that turned out to be key. It's a tenrec from Madagascar. And this study was led by Leah Krubitzer when she was already a faculty member at Davis, but she came back to work on this with me, and an investigator from Germany who had a colony of these. So we were quite lucky. And this is what I would call our roadkill preparation, because the forebrain has been just flattened and cut flat, and that's so you can see all the parts. And it gives you an idea of the proportions of the part. And you say, this olfactory, if the lobster is olfactory, this guy is olfactory. Here's a flattened section through the olfactory bulb. This is the olfactory tubercle. This is the piriform cortex. Hippocampus is folded out here. Neocortex over here. That's septal region, hippocampus, and things up there. So you could come to the conclusion that olfaction dominates up to here. All of this dominates this animal. And it's got a visual area, an auditory area, and a somatosensory area that we can see as being slightly architectonically distinct. And a hippocampus that's at least as big as all of neocortex. So you're going to have some retention of what's happened from day to day. And if we physiologically analyze things, so we have a primary visual area here, auditory area here, two somatosensory areas. Actually, there's a somatosensory belt here and here, and a motor cortex up here. That takes up most of the brain, and the rest we don't know what it does, but you're going to have some limbic cortex on the medial wall, some retrosplenial cortex, which has to do with these areas are really primitive, and they'll have to do with nesting behavior, memories, and so on.
Does this neocortex start to get laminar structure?
Yeah, this has a laminar structure. It's not the best, but I'll have some pictures. It has the laminar structure. So this is just what I told you before, that we need to look for features of brains and see what the features are compared and see what are in common, and you don't say the whole brain is primitive for any mammal. Some features are bound to be unique for every species. And I won't go into that, but the difficulty here is that we're looking at one animal, the tenrec, and it's a member of a whole big radiation of mammals, and it may not represent early mammals that accurately. There may be some marked differences. And here's some of the other animals that would be in Apotheria. And I'll show you an elephant shrew brain quickly, but here's the tenrec. But you can see that they might be quite different in this radiation. And this is just some of the mammals in that radiation. And here's my colleagues at Vanderbilt, including Ken Catania, who got his PhD with me. And here's just bigger brain in an elephant shrew, large visual region, auditory region, and they have at least 2 somatosensory regions. They'd have a motor cortex up here. But let's just go through this radiation, of the 6 major groups and pick some animals and see what their brains are like. And this, in the monitoring branch, you only have 2 groups. You have the platypus and the echidna, and they're quite different. But that's all that's left. And so it's a small fraction, not even 1% of the mammalian species. It's just a fraction. But we're lucky to have them, and people were always puzzled. But you might guess that this is a specialized mammal, and it is. And Leah Krubitzer studied this when she went to Australia, and most of the brain is devoted to representing the receptors of the bill. And it has, this is sort of a proprioceptive region here. This is a tactile and electroreceptive region. Almost all the bill, very little body. The auditory area is squished way to the back, and a visual area way to, so this is highly specialized and doesn't represent really mammals very well at all. It's very interesting because they have divided up this representation into rows of cortex that represent tactile input from the bill and rows that represent the electroreceptive input from the bill. So, and echidna also is thought to have electroreception, but they don't live in water, so it's a little tougher for them to figure out how they use, but it's surmised that they stick their bill into wet termite nests and so on and get some electroreception information. But for the platypus, it's absolutely essential because when it goes in the water, it shuts its eyes, it closes its ears. It can only touch or electroreceptor. So it only has a distant receptor that's electroreception. And so if we went to the next group in terms of branch points on this radiation, it would be marsupials, and marsupials have been called the 7% solution after Sherlock Holmes' cocaine dilution that he used. But it's about 7% of the species on Earth are marsupials. And so, and the monotremes are called monotremes because they have a single opening for defecation, for urination, for birth, and they lay eggs. These guys have a pouch. After about 14 days gestation, they give birth. They don't have the mechanisms of a placenta where they can keep embryo in the uterus for a longer period of time. My wife took this picture. It was on our back porch. We have them regularly coming, looking for handouts. But there's this brain. Some of them made it to the laboratory. And the important thing here is that we've cut off this wonderful olfactory bulb, but olfactory processing remains important. This is a rotated brain, so you're looking at it more from a dorsal, so you don't see how much rhinal or piriform cortex there is. But olfaction remains incredibly important. But you see, we have 2 visual areas. Other cortex is visual, we don't know how it's organized. We have primary somatosensory area, a belt of somatosensory cortex behind it, another belt that's proprioceptive that's right ahead of it, and we have 2 other areas down here, or 2 other somatosensory areas here. This parietal ventral area was first discovered by Leah Krubitzer when she was doing her thesis work, in squirrels that were also caught in my yard. And she's now found it in human brain through imaging, macaque monkey brains, everywhere you look, there are these two areas. And auditory cortex here, which may have subdivisions. But the important thing here, no motor cortex. So if we made a generalization from the tenrec, we would say, oh, motor cortex was there in early mammals. But here we don't have a motor cortex, and we don't see any sign of a motor cortex in reptiles, and we don't see any motor cortex in monotremes. So our conclusion is that the ancestral condition is to not have motor cortex. It's not part of the neocortex of the early mammal brains, and it actually came about with what we'd say is placental mammals, or eutherian mammals, as they're also called. But you can see our familiar guys here, a few visual areas and so on. And we could look at the red kangaroo and other signs of marsupials, but the sad thing is that we know very little about the larger marsupials and how their brains are organized. But there are, I showed you North American opossum. There's animals that look like that are called possums in Australia, and they've been looked at, and the brain is almost identical in organization to the North American possum, even though those lines of marsupials are going back for an extremely long time. For at least 80 million years they've been separated. And the next line we're trying to investigate right now, this is also an early branch, and these are armadillos and sloths, basically. And they're very specialized for what they do. So again, they wouldn't be a good model to say this is what the early mammals would be like, because the early mammals didn't go out with all this armor. They're also known classically as edentates, because that means without teeth. They have little peg-like teeth. That's not, so they're not quite without teeth. But basically they eat termites and ants, and they have a tremendous olfactory system, and finding termites and ants is their big job. And here's what you know so far, but we're working on this brain in the laboratory right now. And you have a somatosensory area, you have a motor area, so this is a placental mammal. And somatosensory regions on each side, a big auditory visual area, maybe other visual areas, but look at the olfactory bulb, the piriform cortex, huge, as would be needed for an animal that finds its food primarily from smell. Now, we're in another group of mammals, Laurasia etheria, and this is a very successful large radiation with a lot of different branches on it. No, we're in eucontiglares, but in this group you have an animal that was once grouped with tenrecs from Madagascar, hedgehogs, but now we know they are not closely related at all. They go back to the very beginnings. They're branching into separate lines. And that's what makes these comparisons more powerful. Now, the hedgehog, again, has some specialized feature of these quills all over it, so you have to be careful in what you infer from it. But the point is that these quills have allowed this animal, an animal of very little brain, as it's been called by like Winnie the Pooh, to survive, because it only has to curl up as in a ball, and it can't be eaten, and then it just has to find food. So it's pretty well off with a small brain. And what?
They're long-lived.
They're
For body weight.
Yeah, because of that,
Yeah.
not because they're smarter. Yeah. But we see here, motor cortex hasn't been demonstrated, but it'd be in this position. Big primary somatosensory, 2 extra somatosensory areas, an auditory area, 2 visual areas, and they're all crowded in together. And then stuff up here we don't know too much about. would be frontal cortex. Some of this would be clearly motor, maybe up to here, further than what I've shown. But this is, we're seeing features in these lines of mammals that have been separated for so many millions of years, and yet they look very similar. And other insectivores, so the hedgehog is one of a whole bunch of insectivores in this group. And Ken Catania has looked at one here, least shrew, which is one of the smallest of mammals, and you can see its brain is quite small on that penny here, and almost a worthless coin nowadays. Copper in it is worth more than the coin. But the point is that you can see a primary motor area here, but you have S1, probably only one secondary area over here, an auditory area, a visual area up in here. And part of the argument is that because this brain is so small, it's actually changed by reducing the number of cortical areas, not increasing, which we'll see that is the pattern generally, but by reducing. So auditory is right next to the secondary area. We don't have a PV in here anymore. We have no area between primary visual cortex and somatosensory cortex. Nothing else there at all. So,
John, if you say visual, this is defined on anatomical grounds, where you say, okay, this wires into a subcortical area.
Yeah.
This is wired to, let's say, the eyes.
so what you can do, you can record, this has been done to see that this responds to vision, and it has a map expected for wave 1. But architectonically, you can look in the flattened cortex and see all these areas. I'll show you some of that. So you can identify it architectonically as well. But for the most part, it hasn't been also identified in these animals I'm talking about by connection. Some of them may have, and some not. Yeah. And so this is another mammal that Ken Catania studied in the same group that I just showed you of the least shrew, the hedgehog. But it's a mole. And a lot of moles aren't so specialized, but this one has this extremely funny nose here, star nose nose appendages. It's just covered with receptors. So this is a specialization that allows this animal to feed very efficiently. And it eats smaller food objects than most other insectivores do. And it can only do this by having this great search strategy, which not using vision at all, but just using touch and flopping this nose all over until it feels something that's important for it, a likely food object. In the same group, or the same superorder, we have bats. And bats and rodents are the most successful orders in terms of number of species. There are huge numbers of rodents, huge numbers of bats. And some people wanted to put these bats because they have a nice visual system, good eyes, fruit bats, because they find their food at night by seeing the reflection of the fruit of the moonlight. They have good smell too. They wanted to put those with primates. It's the flying primate hypothesis of Jack Pettigrew. And that would leave the echolocating bats off by themselves in this order, and these moved over into primates. And they do have some primate-like features. That is that they do have a nice visual system, and primates have a nice visual system, and there are some things that have come about in common by parallel evolution. But that hypothesis has been thrown out completely by the people that are using the genetic methods of determining phylogenies. And those methods give you a lot more to look at, and they're more reliable than looking at how is the visual system organized or something, which you could imagine could evolve in a convergent manner and have resemblances. But here's the flying fox from Rosa, Marcello Rosa. And what you see here is the visual system cortex is in a position more like a primate. Instead of being here, it's rotated and taking up the whole back, and then a secondary area, a third area, and some other visual areas, and say, wow, this looks more like a primate than it does look like a bat. But it shows you what convergent evolution can do. And then there's somatosensory and motor and S2 and PV and so on, other areas. But if you look at, people haven't studied the echolocating bats very thoroughly because they've been focusing on auditory processing, and probably rightly so. But if you just look at the primary auditory area in these bats, they take up, and that's been looked at in a number of bats. It takes up quite a bit of area, and here's two representations which have been shown in the brown bat, but there are other auditory areas all around, but they're more complexly organized. So auditory processing dominates the whole thing, and certain frequencies, the echolocating frequencies, will dominate these representations.
What about the motor area? Don't they have to send out
The what?
motor area to send out the signal?
Oh, the motor area. When, you know, this is a disappointment because they've looked for where somatosensory cortex is. Motor cortex would have to be ahead of that. Doesn't look like there's much room, but how vocalizations are sent out, I'm not sure it's known. I think it hasn't been studied, and it'd be very interesting to do that because, of course, the timing, it has to be connected with the auditory inputs very closely.
Could be tied to the
Yeah,
auditory.
and they have to have feedback from their vocalizations so they can
It could be tied to the auditory areas.
It could be.
There's a lot of subcortical involvement of both colliculus and periaqueductal gray for the vocal production in
Yeah.
bats.
Okay.
Yeah, and the midbrain, inferior colliculus is huge. But my point is that they have small brains, but the echolocating bats, but they don't give you many insights about what early mammals would be like because they've been so specialized. So here's going back and looking at a different opossum. It's a South American opossum, house opossum, because its Latin name is domestica, and you think, well, is it domesticated? No, it loves to move into people's houses like field mice do. That's why they called it that. But I want to show you can identify areas on histological features, and there are different ones here shown. This is flattened brain, so you can get an idea of, in a single section, you might not get all of an area, but you can see that primary visual cortex is very distinct, and this is parvobumin, because parvobumin is used in the sensory pathways from the thalamus. So this is getting direct input from the thalamus. Here's the myelin thing. Sensory areas tend to be more myelin. This difference in shade from here to here is only because the section isn't completely flat. Here's cytochrome oxidase, because an area that is sensory is highly active most of the time, so it'll need more of this metabolic molecule, cytochrome oxidase. There's two of them at different levels there. You can see auditory here and some of somatosensory. So you can identify some areas both physiologically and anatomically in these brains. Here's just if you went through sections. So you can see here, this is calbindin. This is a glutamate transporter, because this glutamate transporter number 2 is not used in cortical neurons. It's only used in thalamic neurons and their terminals in cortex. So you know wherever it stands, it's getting a lot of input from the thalamus. So you can learn a lot about from these things from sections. But if you look at a Nissl section up there, you're not very impressed about how these regions are specialized and differ. Here's from a hedgehog, very old, because you can see one of my first papers. But you can see that the cortex here, this is cortex that would be retrosplenial, which normally has a well-developed layer 2, but a well-developed layer 2 and a thick layer 1 are characteristic, are primitive features. So if you look at all these mammals and decide what were ancestral features, you'll say poor lamination is 1. Here's striate cortex, the best laminated area in the animal. It's not that distinct. Thick layer 1, because layer 1, if you remember from reptilian cortex, there's a lot of inputs coming to basically an outer fiber layer and going horizontal. And you still have a good portion of that in some of these mammals. So a thick layer 1 with a lot of horizontal fibers spreading out, and then well-developed layer 2 in some of these other areas, retrosplenial cortex.
Which is, of course, where a matrix cell still projects.
Yeah, right.
John, you mentioned that the dorsal cortex has relatively few layers
The what?
in the reptile.
Oh, yeah. Basically, it has a single layer of pyramidal cells, and in the fiber layer where these horizontal fibers are coming in, you'll have some of the inhibitory intrinsic neurons there. They're about 10% of the total.
Right. And the piriform cortex, is that similar, or does it have more layers?
So piriform, it's off the scale here, but it has more layers, but not a distinct layer 4.
And what about in the reptile?
In reptiles, it's a single layer.
Okay, so piriform cortex, although it's present in reptiles,
But you can see
has
what,
more
but
layers.
you can see the hippocampus here. Still, it's a single layer of packed cells.
So all of these structures have added more layers, even though they're old in terms of they were present in reptiles. They're present as single layers.
As single layers. So the kind of processing that you would see would be quite different in the hippocampus. The hippocampus, again, is presumably not interested in the pyramidal neurons having a large number of inputs in which they sum and provide an output based on that summed information of a lot of inputs. So it's not very good at making fine distinctions.
And is there any hypothesis how thinking about all of these different cortical areas then increased lamination? There are, and Gordon Shepard has written a comparative study on the lamination of different parts of the brain in reptiles and in mammals, and going from this one-layer scheme. And the hypothesis is that basically it's a retention of these deep layers, and the other things have been added on in evolution. And to get, whether that's true or not, it's hard to answer from a comparative point of view because you have no comparison of an intermediate state. There's nothing intermediate. So you have to have other ways of making inferences, and whether they stand up or not, I'm not certain. Is there anything done about the genetic mechanisms for that?
There are a lot of markers for specific layers in mammalian cortex that can be applied, and some of these markers are now being studied by people that are looking at birds and reptiles to see if they can identify by using these markers. But it's also possible, for example, that you have markers that are specific for layer 4 cells. You would expect them not to be there at all in birds or reptile brains, or in different parts of the brain.
I guess there would be some hypothesis about the developmental process
Yeah.
by which precursor cells
Yeah.
would maybe
Yeah.
have extra multiplications in order to generate cortex.
And of course, if you follow one of the early hypotheses is these extra cells migrated in, and this seemed to be supported when it was discovered that actually in mammals that you have a lateral pathway of cells migrating into cortex as well as the ones generated from the subcortical plate. But these migrating in cells are all inhibitory. So that can't be the source of the layers in the lamination.
But corticogenesis and laminar structure is all, like Colette de Hay and people like that, they have that fairly well worked out in the mammal, no?
In mammals, it's well worked out for macaque monkeys and mice and rats. And presumably, it mainly varies in how long corticogenesis takes place
Right.
and the timing of different components,
Right. Yeah.
but not in the order. But Pashko Rikish did a lot of this stuff on the macaque monkey brain, and he's the strong proponent of no neurons in cortex.
No?
No. But now he has to admit that some come, from a lateral source. But And he said he didn't talk about the olfactory bulb where you're getting the neurons all the time. But his story, if I can tell that take away from my other time, is that you get married, you go on a honeymoon, you come back 3 weeks later, all refreshed. Most of your body has been regenerated in that time. New skin cells, new intestine cells, all over your body, new cells. But not a single one in your brain, all the same. And maybe that's good, so you recognize your wife and you get up in the morning. I'm going to skip some things here. This is just to point out that I've emphasized cortex, but other parts of the brain have changed as well. And if you take two visual animals that aren't closely related, they're related, but not that closely. Tree shrews I'll talk about, squirrels I won't, hedgehogs I already talked about. You can see that here's the dorsolateral geniculate in the thalamus. It's not very distinct from the rest of the thalamus at all. Here it is in a squirrel, very distinct and laminated. Here it is in a tree shrew, very distinct and laminated. Let's look at the midbrain. Midbrain here it is in a hedgehog. This is what early mammals would be like, by the way, this degree of difference. You don't see any real structure in here at all. Look at this nice structure in a highly visual squirrel, highly visual tree shrew, beautiful structure. So things have evolved independently in a similar way in these two lines. Here's a summary, and I'm going to skip that because there isn't much time. But here are the placental mammals, the four major groups of placental mammals, and what we want to be interested in here now is eucontoglires. And eucontoglires have glires, which would be lagomorphs and rodents. Lagomorphs are rabbits and pikas. Over here in this line, it divides up into flying lemurs, which aren't lemurs and don't fly, and tree shrews, and primates. So these would be our comparisons that you want to make to get the ancestor of this group. You'd want to compare across here, and you want to compare across here if you want to get the ancestor of primates. But there aren't many to look at. We could look at a lot of rodents, of course, but that would be redundant. We want to look at tree shrews because flying lemurs glide, but they're really not available for study. I've looked at some of the brains, but that's about it. So here's a tree shrew, which LeGros-Clark was classified as a primate because of similarities to primates, but again, the same mistake because he looked at the visual system, and the visual system, probably by convergent evolution, has many resemblances that would fool you into thinking this must be a primate. But they don't look very much like a primate, so we have other reasons. But genetically now we know they're distant enough that you wouldn't put them in there. And the brain isn't very much like a primate brain either, in the sense that we pride ourselves on our frontal lobes. And frontal lobes are for planning, thinking ahead, weighing the consequences of your behavior. And this would be an animal that doesn't weigh the consequences of his behavior very often. The main thing it does is run away. It's got to be fast, but it is visual and a visual predator. And this is one of my students' summary of brain organization built on other studies, but he really studied the motor cortex and so on. And what's there is some visual areas take up this huge amount of temporal cortex, so it is highly visual. Auditory down here. There are usual somatosensory areas here. This is showing different body parts. Forelimb here. Face is always large for almost all mammals, large face representation, because they use their face in motor activity and use their face as a sensory region. More than forepaw is unusual. That comes in with primates. But one of the points here is that the connections to motor cortex come from visual areas, and this is a very little region that we can call posterior parietal cortex, which becomes huge in primates, and I'll want to get to that. But here's an anatomical experiment with an injection in the hand representation in somatosensory cortex, and there are two somatosensory areas, mirror images of one another here, and here's a joined hand region, and the connections match. Good. But nothing direct to motor cortex, instead to this proprioceptive area here and to this other somatosensory belt region here. Very few connections back here. And here's, in contrast, the injection into motor cortex. And now we see that we get lots of visual inputs directly to motor cortex. Not directly somatosensory. Indirectly we get somatosensory, but a lot of visual inputs from these visual areas and also from this little bit of posterior parietal cortex. But posterior parietal cortex is not the main source of visual information.
Is that the visual thalamus you're injecting?
What?
You're injecting
Motor cortex.
oh, sorry.
Motor cortex. So where's the motor cortex getting its information from? It's getting its information from visual areas directly. There's one similarity in the visual system that suggests that tree shoes did bring something. Their ancestors of tree shoes and primates did have something in common, although this is still a question that could be parallel evolution. And those of you that know visual cortex organization know that cells there represent the orientation of a stimulus. And the cells that are activated by the same orientation in primates are grouped together in primary visual cortex. And this is a color code for that grouping done by optical imaging here. And also connections were studied, and cells that have the same preference will be selectively interconnected. This is from David Fitzpatrick's study. But the point is that tree shoes have this kind of cortical organization in primary visual cortex. All primates do, and rodents do not. Even squirrels with a highly developed visual system do not have this. So it wouldn't go back into an ancestor of the whole group. Eucontigula's ancestors wouldn't have this feature. It's not a feature common to mammals. It's an unusual feature for mammals. But all primates and tree shoes share it. So we could say it's likely that this developed before that breakup occurred between those two lines of evolution.
So there's evidence that rodents may have an orientation.
They have orientation selected cells, but they're not grouped.
In the barrel cortex.
Oh, in the barrel cortex, yes. Yeah.
So it could
Yeah.
be a property of the way the cortex surface can self-organize themselves.
That's at least true for rats, and I don't know if it's been demonstrated in mice, but I would guess it'd be there. But squirrels are highly visual and aren't using their vibrissae to identify objects like rats and mice are. So I'm not sure if the rat system would even hold for all rodents.
If you could find a strongly visual rodent, you might predict that it would have orientation
Yes.
cells. I don't know.
But the squirrel is a highly visual rodent.
Right, okay.
You don't get any more visual than that in rodents. Yeah. What?
What kind of organization do you find in the squirrel case, for instance? Because let's say the cortical projections already impose some constraints on how this can organize
right.
itself, so it will not be random.
No.
So it might not look like this. So what structure do you find?
The property of orientation selectivity is thought to be created by the cortical wiring, the selectivity, although some people argue there's a bias from the geniculate cells that's already starting with the bias, so retinal cells. So they could be grouped or not grouped depending on the kind of intrinsic wiring that you would get.
So there will be a group
This
that will look different.
Yeah.
Okay.
So you would get the homogeneous color because the individual cells are randomly distributed and they're not selectively interconnected.
So no
Regions aren't. Cells might be selectively interconnected, but regions are not selectively interconnected. So I won't go into a summary here either because we're running out, and I wanted to just go through a couple other things very fast. Not even this, but early primates were nocturnal, small. They had to adjust to fine branch living, so they had to have coordination. They had to have eye-hand coordination. The snout is reduced, large eyes. They're not going to grasp food with their face so often because that endangers their eyes. It's safer to grab it with your hand. And of the mammals or primates that exist today, we have some representatives that look a lot like the early primates. Their brain shape is highly similar, although somewhat bigger for the body size. This would be the African prosimians. Galagos would be the least specialized out of the prosimian group. And so not your typical looking primate, but they are nocturnal. And we've studied them a lot. And I'm going to just go quickly, but you can see there are a lot of cortical areas, and we can start to identify these areas in the visual system that are specific and only have been identified in primates. Like MT over here, this collection of areas around MT which deal with motion analysis have all been identified only in primates, although it's possible that other animals might have them. Posterior parietal cortex is huge. You can see this huge area, PP, over here, and I'm going to talk a little bit more about that and very quickly. And you have modular components, cytochrome oxidase blobs. All primates have them. None of the non-primates in the whole eucontiglaires have this feature of suborganization or banding in V2. There's weak banding here. So there's a lot of things that I could talk about. But I want to talk about some subdivisions that are functionally related of posterior parietal cortex and the adjoining somatosensory cortex that have to do with grasping behavior, defensive behavior, and reaching behavior. And the reason that we know that it has that behavior has to do with electrical stimulation and other studies. But I just want to point out that these two areas can be defined architectonically in fat cortex. This other region around here also can be defined. You probably can't see it because it's faint, and so on. So we have, here in these cases, we have microelectrode mapping data, anatomical connection data, architectonic data to support these kind of conclusions. Are there three visual areas? V1, V2, V3. This has been controversial till about just a few years ago when the brain I showed you figured out that how to answer that question. And the question, to convince people, was answered anatomically. An injection was made here. It seems very simple. The question is whether there's a third area out here. People thought there was an area up here, but not down here. So if you inject here, you get label here, and you get label here. There's three areas, right? We also get label here, it's fainter, and we get label over here that's fainter. But you can't see unless you go to higher power. So we know what the main connections are very easily. But I want to talk in just 5 minutes about Michael Graziano, who introduced me to a different way of looking at the organization of cortex through electrical stimulation. We'd used electrical stimulation for many years, but short trains of electrical stimulation which invoke a little tiny movement like a finger movement. So you can map motor cortex that way and get a somatotopic or a homunculus kind of map of motor movements there, and in premotor cortex and so on. We use that quite a bit. But what he did was look at the traditional motor areas, but in a different way, and I won't go into their somatotopy, looking at longer trains. And this was used in Spain a long time ago. And I don't know if you know these experiments, but they're really old. And Diego was quite brave here because he's standing here with not a sword, but a radio transmitter. And there's an electrode buried in the bull's brain. And when the bull is charging him, he turns it on and stimulates its brain, and the bull changes his mind about what it wants to do, or at least it stops and doesn't kill him. He tried it out on graduate students till they perfected it. And when Graziano used long half-second trains of electrical pulses in awake macaque monkeys and motor cortex, he got more complex movements. So you got defensive movements here and hand-to-mouth movements here, hand manipulation movements over here. And that intrigued me after I saw this. I said, well, can we use this to investigate other parts of the brain? He also looked at a region in posterior parietal cortex and found he could get defensive movements there from a region that's been defined before. And this is what a defensive movement to an air puff looks like. So the monkey gets the air puff, it's noxious, and it shuts its eye and grimaces and puts its hand up to protect its face. So, and these are my two coworkers who worked on this with me, and I have no time. So we looked at posterior parietal cortex and anesthetized galagos, and we could do this and get the movements in it. So the animal isn't thinking, I want to make a movement or doing anything. It's anesthetized, but it still does it. You got face defensive here, forelimb defensive here, hind limb and forelimb together as if climbing, forelimb reach, hand to mouth movements here, forelimb face, face aggressive movements here, and grasping movements up here, which came in a later experiment, so it's not on this slide. And this is just some of the movements. I won't go into it, but here they are. We've found these three main movements and other movements, but we've concentrated on looking in New World monkey, grasping here, defensive reach, because they're pretty reliable. So it's not just in prosimian primates. And here's a squirrel monkey, same three regions we've identified. So two different New World monkeys. And in macaque monkeys, we haven't done that much, but we got a face defensive region here, a grasp region here. Graziano found a defensive region here, and other people have implicated this region in reach as well back here. So you have this reach, defense, grasp in the same sort of spatial relationship in all these primates, and we don't get anything like this in rodent or in a tree shoe and so on. We don't even see much posterior parietal cortex. So posterior parietal cortex has become an important part of the brain. It's an important part in all primates. This is a part that's especially enlarged in the human brain, would be used for all kinds of tool manipulations, all kinds of different tasks that are specific to humans. But we have regions for ethologically, I would say, relevant behaviors pre-wired into the system. And of course, this system works through motor cortex. So if we put a block, a muscimol block in motor cortex, you can't get the behavior anymore. We know where to go. Yes?
So these behaviors are, because they are in the cortex, they are probably learned.
Well, if they were learned, I'm not saying that experience would modify them, but they're always in the same spatial relationships, and they're in spatial relationships across primate species that have been separated by millions of years. And I'll go back to we were talking about the lobsters. You're going to have some behaviors that you can't really learn, because by the time you learn them, it's too late, you're dead. And a defensive posture or an aggressive posture would be things that you would think you can modify by experience, but they have to be sort of pre-wired. And so we would think these same behaviors would be in other animals, but having centers for controlling them in posterior parietal cortex wouldn't be in non-primates. This is a new addition added to the machinery.
I do find this very surprising, very interesting, and I'm reminded of Wilder Penfield, because he was doing these stimulation studies in humans. Do these methods you're talking about differ significantly from what Penfield did?
They use surface stimulation, which is harder because it's hard to get enough current to get the pyramidal cells that are providing output, and shorter times. But Sherrington using surface stimulation and in the early, 100 years ago or more studies, when they did their maps, they said, this is the map of the first movement. If we continue to simulate, we get a continuation and a more complex movement. But people were not interested in the more complex movement. They were interested in the first movement, because for a neurologist, that would give them some localization information.
Here there's some degree of coordination. It isn't just a simple movement.
It's a movement that takes about, the time course of these movements is about a half a second. And we have information on the time course and so on. We have information on connections. We also have optical imaging. When we're electrically stimulating, we can see what other parts of cortex are activated. Because our criticism is if you look at the connections, you'd say, well, you're connecting to other parts of posterior parietal cortex. Everything must be going on. You don't have an idea of what you're doing. But if you use optical imaging, it appears that the connections to other posterior parietal cortex it goes no further. It dies there because we think it's selectively on the inhibitory neurons, and it's to make a decision of what behavior out of the arrays of behavior that you can have should be made. So they're mutually inhibitory. And if you start one, you kill off the others. What leads you to start one and not another, we would guess would be not only sensory inputs, but prefrontal inputs as well.
So John, are you saying that this is the complete set of these stereotype behaviors that would be...
These are the behaviors we've looked at, and I should say if you look, you go from face aggressive to face +4 on aggressive behaviors, but as you move the electrode around here, you get that category, but it changes with location. The exact form of it changes. The reach movement might change where the reach is directed, for example. So within there, there's sub-organization. And we've only done one experiment where we tried to train the animal with reaching hundreds and hundreds of times a day, and we thought that we could enhance this kind of behavior by that training. Whether that really is true or not, I can't say from one animal.
But then you also have much more primitive centers that are very much engaged in controlling the stereotype behavior. I would think it's
Yes,
brain,
yes,
for instance.
yes.
So is this part of the motor system really interacting with areas like the central brain?
So yeah, the older areas are there, and I have a nice postdoc now from Estonia who is not afraid of hard things. And he's going to be looking at the subcortical connections because these would have to link in with those appropriate centers, amygdala and other places as well. But he's just going to look at the defensive circuitry in more detail. But you're exactly right, because these would be an add-on, in a sense. The posterior parietal cortex would be an add-on to a system that was already there, and largely the specifics of it would be mediated subcortically, I think.
But you could imagine
Depending on
also
what the behavior.
civilization, so that the organization of these behaviors migrates from the midbrain to the cortex, in the same way that some of the visual functions of the visual midbrain have migrated to cortex.
So you can have functions that were completely subcortical and then no longer can be mediated solely subcortically. Yeah.
If you were wildly speculating, would you think that in human higher cognitive function that there might be some, I don't know, like say, in the same way that reaching is something we better know how to do more or less before we're born, maybe certain kinds of thinking or language.
Anybody there's two things that babies do. They reach and then they put things in their mouth.
Yeah.
So that's pretty basic at an early stage. But those posterior, in functional imaging studies, posterior parietal cortex regions seem to be heavily involved in tool use or even thinking about tool use. And so we have so many abilities that depend on extending our body by using tools that chimpanzees do to a limited extent, but most mammals do not use tools in the same way we do. So this would be a pre-adaptation just by good fortune for moving into tool use.
Okay.
Yeah.
Just a comment about rodents. So in the motor cortex, in the bristle region, there is a small area. If you stimulate, you get whisking, regular exploratory whisking, and then there's a larger area. If you stimulate, you get whisker retraction movements, which could be defensive. So it could be
It could be. And there's a report of getting defensive movements by stimulating the tectum in rodents. So there are other places that we need to consider. I'm not even going to summarize things here because I'm out of time. All I can say is this is a souped up version of the macaque monkey thing that I showed you before. Didn't have time to put all the label on here. I can find 100 things wrong with this, but if you take this at its face value, there are proposed 90 areas, 98 areas in in macaque monkey brain. It's speculated that human brain would have over 200 areas that were functionally distinct.
You're talking about
It's very-
vision alone?
No, the whole thing,
The whole brain?
the whole thing, yeah, not just vision. Vision stays at, and now can I just have 5 minutes?
As far as I'm concerned,
Anybody against that? So I just want to talk about another way we can learn about brain evolution, and I don't have time to really go into it, but smallest primate, largest primate brain, that's the relative difference, not the absolute. And so a lot of people have thought about scaling as a way of scaling rules, developing scaling rules and thinking, what do you have to do when you go from a large to a small brain? And this is to just demonstrate that in our imagination we're fascinated by this Dali picture because we know that the elephants could never walk on something like this. We know it's impossible, so that we're intrigued by things that are unusual or seem to be impossible. And this is just that neurons, as they scale up, gain bulk in their axons and in their dendrites disproportionately, and creates a problem of space in the brain, especially for long connections as they get longer. And this is just that if cortical areas get larger, it's by making changes in the dendritic arbor, it's harder to change what they do. Small areas can change what they do by the smaller changes in the circuitry of individual neurons. So smaller areas in principle would be more valuable. Large areas in principle can only look at details of sensory or other kind of inputs, and they wouldn't be very good at global processing. So some of the largest neurons are in prefrontal cortex, and the smallest neurons, whether they're dendrites or intrinsic neurons, are in visual cortex, primary visual cortex. It's good for detail, but you wouldn't live very well with it. This is just from Elston's showing the dendritic arbors in different areas and how they look like from the surface view looking down. And you say it's smallest here for sure. There's a progression because the functions change with the size of the area and what they're good at. And primary visual cortex in a human brain and in a macaque brain, if you flatten them out and measure the surface area, are the same size. Even though our brain is 3 times bigger, we haven't increased because it'd be pointless to do this. But this allows us to worry about a wiring problem, and the wiring problem can be changed in a large brain by making more areas and making modules within areas and having processing locally, inner hemisphere specialized, few connections between the hemispheres and so on. I can't go into this, but I just want to say one thing about Susanna Herculano-Huso, who introduced us to a practical way of determining how many neurons are in any structure of the brain. And that seems counterintuitive as to count the nuclei, not the cells, because the problem with counting cells is that you have to identify them histologically in sections, and the brain is not homogeneous, but you can basically put brain parts in a blender and centrifuge out the nuclei and count the nuclei. And you can do this in a flow cytometer. That's how we're doing it. You can automate the process. So you can find out, for example, this is one thing with Susanna, that for roughly the same size of a rodent brain and a primate brain, almost the same size, 1.5 billion neurons, 9.9 billion neurons.
Distinguish the neurons from glia?
What?
The neurons from glia?
From
Glia. Glial cells.
glia. Oh, you can distinguish the neurons from the glia by using 2 different stains for different proteins in the nuclei. So nu-N will give you neurons. Yeah. So yeah, and I'm not talking about the other cells, but that's an interesting story at all. Well, the point here is that all primates have more neurons than any other animal for brains of the same size. And that means that the neurons on average has to be smaller. And if we were going to have the number of neurons in a human brain in a rodent, it would have to be an unbelievably huge rodent, and it still wouldn't get to that number. So here we have roughly 100 billion neurons, someone less than this. This hugest rodent that ever lived would only have 24.8 billion neurons, based on our scaling for neurons in rodent brains looking at a whole range of neurons, of rodents, and a whole range of primates. This difference is huge. And here just summarizes that. Elephant, chimpanzee has 6.5 billion neurons. An elephant only has 10 billion neurons, but look at it's got this huge brain. But we have so many more, 20 billion roughly. I won't go into that. I'll just end. It'd be fun to talk more about that, but I would point out that this is an inferred behavior from footprints that left in ash, that two individuals walking were sexually dimorphic, one bigger than the other. So the assumption was that one walking there 3 million years ago was comforting the other one. And same thing happens today. So we retain some of these behaviors. But some people would call this mate guarding. Thank you very much for your time.
We went through all these maps, and I think it's very amazing how this area started piling up. But for me, it's now hard to synthesize what are the kind of rules or the functions, or what are the principles you could find through evolution that would apply.
So, one of the things that I think is so amazing about primate brains is that there's enough evidence that you can say that the number of areas from early mammals, which would be on the order of 15 to 25, in that ballpark of areas, now is clearly on the order in a macaque monkey of 90 to 100, and probably in a human over 200. So there's been an increase. We don't know that's true for any other order of mammals. We don't know that there has or has not been an increase.
How many in a rodent?
So in a rodent, you're going to have an order of 25, 30, maybe, but nobody has looked at a large rodent. So the rat is the only one that's well understood. And so it'd be great to look at a large, but it'd be interesting to look at a lion brain to see if any carnivore has quite a few auditory areas. It has a number of visual areas of the ones that have been studied. Cats the most. But we don't know if there's any difference in a large carnivore like a lion and a domestic cat, which has been studied extensively, or ferrets now, which have been studied quite a bit. So these are big gaps in our understanding. But it's pretty clear that in primates we've taken advantage of what becoming more modular. As the brain gets bigger, it has more parts, and so processing can remain fairly local for most of the things that are done. And then only a few connections would be fast and long, because as you get a bigger brain, the real problem is connections have to be longer. That's time. The only way you can compensate for that time is to make the axons very thick, and that's space. And you know, somebody said if we wanted to do this and just scale up a small primate brain to a human brain, increasing the number of neurons and keeping the connections the same proportionally, our brains would be about the size of a bathtub, just from the space of the connections that would be needed.
So, but it's quite interesting in this sort of evolutionary study, the emergence of these structure-function relations. So you show that an animal that uses vision has big visual areas, animals that have other modalities that they're using, we see specialization. And I'm wondering about the same process being applied to increased laminarization. So when we see this creation of new areas, it's kind of easy to make the structure-function relation. But what about what function do we gain by having increased laminar structure?
I think what you gain is that you get another dimension of specialization. So you have a crude specialization in all mammals in neocortex, not all areas of neocortex, but of 6 layers. And you could say layer 6 provides feedback to the structures that are providing the input. Layer 4 gets the detailed input. layer 1 and other layers get other kinds of input that are more modulatory. Layer 3 projects to other area of cortex. Layer 5 projects subcortically. So on the basis of inputs, outputs, you've got that big category. But then you can start to subdivide, and you can keep things separate and specialized. So layer 4 can be in as many as 3 or 4 sublayers. So in one animal, you might separate cells that respond to light onset from cells that respond to light offset. That's unusual, but it's done. Others you will separate what's called the magnocellular stream from the parvocellular stream. So
Within a
different layers.
Okay.
So they'll be one over the other, separated. And then their connections can be mixed or separated at subsequent levels.
So it's kind of a
So you're starting to get more subdivisions. And it's similar if you look at a bird of prey like a hawk or an eagle, the lamination of this optic tectum is fantastic. If you look if you look at the primary visual cortex of a tarsier, and I have slides, it has more subdivisions of primary visual cortex, which is a third of all cortex. It occupies a third of all cortex, and it's subdivided all this way. And they're visual predator specialists. They don't do much else but detect prey and grab it.
How does the brain integrate different sensory modalities?
That's a good question, and I think in all the animals that we talked about, there's an integration. And the integration can, instead of coming sooner in a human brain, I think comes later. So in a mouse or rat brain, primary visual cortex will go to motor cortex. Somatosensory won't go to motor cortex. Maybe auditory does as well, I don't know. But you would have your motor functions influenced by this direct primary sensory inputs coming to these regions. In a larger brain, you will analyze the visual information and the auditory information further before you use it to influence the motor and do multi-sensory processing.
They come together.
But there are some areas that are seen to be specialized in primate brains for multi-sensory processing and get multi-sensory areas inputs right away. And one area is a second level auditory area called CM. And it gets somatosensory inputs and auditory inputs. Auditory inputs are at a second level of cortical processing. Somatosensory maybe at a third or fourth level. But it's not clear yet why you want to mix somatosensory and auditory at such an early stage. But in an opossum, the parietal ventral area, which is the second level of somatosensory processing, also processes auditory information. So there's something about auditory and somatosensory that mixing will be done in a different way in different species, but it's done very early if you only have a few areas, but even in primates it can be done fairly early.
And related to that, how does the brain then, how does a different sensory area recruit an area that's been specialized for something else if that area is not stimulated?
How do the connections patterns change in evolution?
Yeah, say I'm blind, and
okay. Yeah, there's been quite a bit of research in that, and my former student Leah Krubitzer has done some of it, and just publishing recently of having animals raised blind or raised deaf. And what you first, what I haven't mentioned at all is many connections in the cortex, especially, are what people have called exuberant in development. The brain in general has what for a long time is called regressive events. So you have too many neurons, you have too many connections, and you lose them in development. And the crash can be tremendous, up to 80% in some structures of neurons lost, but more likely you're losing connections in neurons. So if you change the rules of the game and remove an auditory input or a visual input, you remove a competition. And so you will have visual areas becoming auditory or auditory areas becoming visual. And the interesting question, which is hard to answer, is If an auditory area is responding to visual inputs, is it seeing or hearing? I mean, what's the phenomenon like in that case? But we know in humans, the Braille readers, that they depend on primary visual cortex for the Braille reading. And there's an unfortunate case of a Braille reader who was very proficient and taught it, had a stroke of primary visual cortex, and she couldn't read Braille anymore, and she couldn't relearn it. And obviously, she couldn't teach it anymore.
Thanks.
Paul, Aaron?
So we see this laminar structure of cortex emerging, right? And now on the one hand, there are these suggestions that across this lamina, you might have, let's say, what some people call a canonical kind of micro-range or microcircuit that would be sort of repeated as a template throughout the whole sheet. Do you buy that?
I buy that, but I would say that the details of that circuit are modified according to the functional column within an area and the area. So the details are modified.
But is it modified to, let's say, 50% or 80% or 10%?
Well, you can modify in a lot of different ways. You can modify, if you're just talking about inputs and outputs, structurally you don't need to modify the column at all. But you're modifying the functions tremendously by inputs and outputs. But some areas probably have more modulatory transmitters that would allow them to be more plastic, and some layers more plastic than others, more subject to change. Some would have smaller neurons because those functions would be more important than larger neurons. The dendritic arbors would be different. The pattern of the intrinsic connections of how regions interact with one another. So it's sort of sub-layers, with specialized cells, neurotransmitters and so on, sub-layers. All these things are variables.
Orthogonal to the lamina, you might also see different kinds of clustering. As also shows for V1, for example, you could think about the barrel cortex, or you could think about, let's say, blobs, for column, and so on. So is that also a design feature, that you get sort of different kinds of clustering of cells?
So that kind of clustering, and it can be a sharp boundary or gradual. So if you think of something that's continuous, like tone frequency, in auditory cortex, you wouldn't expect to see boundaries, but you would expect to see an arrangement. But this seems to be a basic feature of nervous systems, that they group and segregate inputs. And the good evidence is really old, but I like it the best is from Martha Constantine Patton, where she put in a developing frog larva, put extra eyes. So you make a little slit and put an eye in. She could get optic tectum, get input from 2 eyes instead of 1. And they both want to go to the whole tectum and occupy the whole tectum, but they're incompatible with one another. So they have a chemical signal, so they go to roughly the same pattern. But locally, they form stripes. They segregate completely, so you don't get 2 eyes. You get 1 eye stripe, next eye stripe, over and over again. So you either, depending on the dynamics, and it's been modeled, you get either stripes or you get a dot and surround, depending on the magnitude of the 2 inputs for any of these kind of things. The dot and surround would be the ocular dot to the cytochrome oxidase blob kind of organization, for example.
Right.
The stripes would be what you have in V2. And those are things that are, or ocular dominants are usually stripes. So that's my way of looking at it. Yours too?
Yeah, no, it makes sense. But you have sort of a competitive system in which you organize the response properties of these neurons. If you've done some models of that.
So what I liked about her data is that the frogs never had binocular input, you know, their ancestors never had. And yet the nervous system treated it just like you were in stride cortex or someplace where you would have 2 inputs in competition with one another.
Okay, Aaron?
I have a question about different kinds of functional differentiation. There's the kind of functional differentiation that you've been talking about, for instance, something concerning vision or motor control or olfactory. And those blackboards and mobs of blackboard and vertical walls and horizontal ceilings and so on, look in another direction, I see something totally different. Quite different regions are significant differences in sizes, their contents and so on. I need to integrate them as well as cope with the fact that they're new ones. Now, that suggests that there are ways in which brains of animals that have visual functions like ours can do very rapid reorganization of function. That clearly can't be done by changing the wiring, so it must be done in some other way. Is anything known about how that is done? I mean, computer science would say there has to be virtual machines and not physical machines.
I'm not sure how it's done, but I can tell you what some views are. And one view, which would be from Wolf Singer, would be a person that would promote this, is that you need a different code for those kind of things. So you have basically a rate code for where is something, how intense it is, these simple sort of things. The neurons fire more to the stimulus or less. That rate code is fairly stable. So they're basically arguing that the correlation of activity of neurons in one location in the brain and another location in the brain is another way of getting information. So, and that's saying these things belong together or these things are part of a domain, but it's a different kind of code. So the two codes are operating simultaneously. The correlation code is fluctuating rapidly in microseconds, and so some brain states are changing rapidly as a result. Whether that's the answer or not, we do need an answer to that kind of question.
And you have to be able to cope with many different sorts of change because you're seeing not just different patterns in the visual field, but different kinds of things like there's an animal, or there's a tree, or there's a rock, or there's different kinds of material, water, clouds, and so on.
And people working on vertebrates, I think, have made some of the best progress. I'm not really that familiar with that literature in terms of how circuits have a dynamic nature and what they do can change very rapidly. And that hasn't been investigated that effectively in mammals.
I think we need to give our speakers some lunch. But one last question over there.
Yeah, it's a question related to when you make this comparison with the power station where you cannot unplug all the equipment. How is this how much is this a concern for the evolution of the nervous system? If there are structures that, I mean, is evolution able to disconnect structures that are not functional anymore, or do you have any example of that?
Well, what would happen, and there are examples. For example, there's so-called blind mole rats. And they're not blind, but their eyes are covered with fur because they live in tunnels and you don't want to get dirt in your eyes. So they're only dimly, they're photosensitive, but they don't form object vision anymore. But vision is still important because you have to know the annual cycle. When to breed is very critical. Circadian rhythms are very critical. But their striate cortex is still there. It's a tiny sliver of cortex. It's not basically non-functional and responds to auditory inputs. But architectonically, it can be identified. It has connections with a little tiny lateral geniculate visual nucleus. It gets visual inputs, but it doesn't. Visual cortex is basically gone.
Okay, well, thank you very much.